So again, just to indicate a few things to you, these next few slides are ones that I sort of stopped at. You do need to know about the different types of glands because they will be on the term test, whether they're part of multiple choice test questions or a one-line answer. So which are the types of glands that produce earwax? And you'll just have to be able to write that out. That's an example of a one-line question, and you'll see the exact same types of questions as practice on Thursday. So we had talked about these ceruminous glands, and we had talked about their function, and we had sort of left here. I hadn't really gotten into the sebaceous glands, but you do need to know these sebaceous glands as well. A good case in point, where I have these different bullet points up here for you, and I've covered them, it's probably a good idea to make sure that you know everything that's mentioned here on these different bullet points. So these sebaceous glands are a little bit different than the glands that we've been looking at previously. They're over the entire body, except on the smooth skin or the glabrous skin. So anywhere like your palms, for example, you would not have these sebaceous glands, nor on the soles of your feet. That's glabrous or smooth skin. And again, that name not being too important. The reason why we call these sebaceous glands is they produce that substance that we talked about all the way back in lecture 1. When we talked about acne, we had talked about sebum and the production of sebum and how bacteria digest sebum and use it, and how that actually causes their growth. And this sebum that we've been talking about is from these different types of sebaceous glands. So you have them on your face, for example, and this is actually an oily substance that is caused by the breakdown of the cellular membranes. And it's oily because of the lipids that are found in your cellular membranes. So again, I don't know how much histology all of you have had, but these are classified as simple alveolar glands. And like the alveoli you have in your lungs, an alveola or an alveolar gland just means that it looks like a sac. So it's shaped like a sac, just like your alveoli are shaped like sacs. These alveolar glands you can actually see sort of off to the side. They kind of look like they have a shape of a round sac or like a balloon shape. And these are also the types of secretion that you get are known as holocrine secretion. So the glands are consisting of these different cells. Within the gland, these cells start to break up, and as these cells start to digest, parts of their cell membranes are digested as well, and they will then be part of that secretion that is pushed up from these alveolar glands. So this is different, in other words, from the miracrine sweat glands that we talked about last week, which are more like classic exocytosis. They are releasing substances packaged into the vesicles. Here, the entire cell is dying off. It's being digested. It's forming that lipid breakdown product, which is making up part of the sebum. Most often, we find these little sac-like glands, these alveolar-type glands, associated with hair follicles. So in that sense, it is similar to those apocrine glands we looked at before. And the function of sebum is to really collect dirt. It's also to soften as well as to lubricate the hair as well as the skin. So it does have its normal function, but when we have certain types of bacteria, they will actually use these secretions to help them proliferate and to dominate. If I don't talk about something on this figure, you don't need to worry about anything on that figure that I haven't mentioned. Okay, so why are we talking about all of this? And why did we sort of take a little tangent into skin repair and what happens after skin injury? And we were hypothesizing that in terms of the new model systems that we're looking at, can cancer be initiated by bacteria? So it's taken for granted nowadays that viruses and viral infections, for example, when we talk about different types of HPV and we talk about cervical cancer, that's now well established that viruses invading and infecting cells can actually cause cancer. But can bacteria cause cancer? And that's one of those really interesting hypotheses. And over the last 5 years or so, since about 2015-ish, a number of groups have actually suggested, and this is one of those, and this is a Nature Communications article, is that maybe when you are looking at a wound on your skin, and there happens to be bacteria, and there happens to be an immune reaction to that bacteria, that this could actually initiate skin cancer. It was very, very controversial, to be quite honest. And Fiona Watt is actually a very controversial figure. If you are taking different immunology classes, you may hear about certain types of cells. So going back and trying to tie in what we started off looking at in the last lecture and what we have been looking at throughout this course, we've been looking at the different layers of the skin, particularly the epidermal layer. And we've been looking at different forms of cancer, whether it's a melanoma, a basal cell carcinoma, or a squamous cell carcinoma, and how they originate in these different cell types, and how theoretically they can actually infiltrate into deeper layers. Some of the other things that we've also been looking at along the way is what happens when you damage your skin. So when you cut your skin, for example, it has the capacity to heal itself. And one of the things that we talked about was its ability to repair after this initial injury. And one of the things that we haven't talked about, although we took a side tangent, was associated with this skin repair, where we have a lot of fibrinogen and we have an overactive number of fibroblasts, we can actually develop what is known as a keloid scar. We get much more scar tissue than we would actually anticipate. It goes beyond the edges of where the skin damage has actually occurred. So your skin, in certain instances, actually has the ability to proliferate more than normal. And so if you look at, for example, this initial injury, it can get completely healed. You may develop a small scar, similar to what we talked about. However, if you were to continually damage that area of the skin, or you have continued damage to this area of the skin, even though it might begin the process of healing, you might actually develop what is known as an ulcer. And today, we're going to be going back and looking at if you have an ulcer on your skin, where you have a scar that doesn't completely heal, and you happen to have bacteria on your skin, could that result in not just Marjolin's ulcer, but could it also develop into skin cancer? And that's, again, a really interesting and somewhat evocative hypothesis. So just again, to go back and maybe get you to think a little bit about this, we typically kind of bypass the fact that skin is actually a really rich source of a number of different immune cells. And we talked about that very briefly at the end of lecture number 2. We know, for example, when you damage your skin, if you go in for surgery, one of the things that often happens, even in the hospital, is what is known as a surgical site infection. So you'll go in, you'll get some kind of operation. They will use sterile procedures, but regardless of what happens when you go into the hospital, you might develop a surgical site infection. In fact, it's very common that surgical site infections are actually a source of concern. And one of the things that often happens, especially in surgery, your immune system function goes down. Can you think of a reason why? Any ideas as to why, if you're going in for general surgery, why your skin immune system actually might go down and why you might actually develop an infection at the site of where you're getting the surgery done? Any ideas? What happens when you go in for surgery? And I think 1 or 2 of you had indicated that you might want to actually shadow these individuals. Any ideas? You're going in for general surgery. I'm going in for an appendectomy tomorrow. Yes? So you might have to undergo general anesthesia. So you are inhaling or you get an injection of a general anesthetic. So what's wrong with that? Any ideas there? So one of the side products of having general anesthesia is that your immune system, the immune function, actually the neutrophils among the ones that are most affected, they actually become less active. And so your immune system during the time you are under general anesthetic is actually depressed. And the reason for that is because you are actually cold. So during the time that you're administered an anesthetic, it actually affects your hypothalamus, your core body temperature drops. As your core body temperature drops, many of you will know that your immune system is less active and it's not functioning quite as well. And your neutrophils, those surveillance immune cells, which are also capable of infiltrating the skin, become less active as well. So when your mom tells you that if you're cold, you are more likely to get sick, she's kind of right, in the sense that in surgery, having a drop in body temperature will actually lead to a suppression of your immune system. We also know, for example, and we'll hear about this from some of our guest speakers after the term test in the second half of the course, that if you are given an immune suppressant, so you have to have a transplant of some sort, that one of the things that happens as a result of having an immunosuppressant, so you don't reject the tissue that's being transplanted, is that you will actually increase the rate of skin cancer. So there is a link, in other words, that I'm trying to make between the immune system that's part of your skin and affecting it, as well as producing things like cancer. And this is, again, where it becomes very, very controversial. So there is also some evidence, and increasing evidence. Not everyone believes this, by the way. So Fiona Watt, one of the authors on that publication that I showed you earlier, is one of the big proponents that the immune system and specialized cells in the immune system are involved in the initiation of cancer. But what we've been looking at up till now is the skin microbiome. Everyone has a unique skin microbiome. Women and men have different microbiomes. We can genotype this skin microbiome as well, and this resident bacteria might, in some instances, play a role in initiating this cancer because we have so much of it on our skin, and if we can somehow affect the immune system or alter the immune system, then maybe this is a way that we could actually cause skin cancer. So this link between having a wound on your skin, having some kind of inflammatory response that gets triggered as a result, and developing cancer is one of those hypotheses that has been kind of out there for a little while. One of these, for example, that kind of makes that link feasible and why this might actually work in practice is that if you look at that situation that I talked to you about earlier, some individuals have this propensity of developing what is known as Marjolin's ulcer. So you get chronic skin damage all the time. That skin never really fully heals 100%. And these types of ulcers, where there's skin damage, really, it tends to be raw all the time. For those individuals, they develop certain types of skin cancers like squamous cell carcinomas. So this link that maybe that squamous cell carcinoma on your skin, because it's not really healing well, on an area that's been damaged over and over again, might involve those bacteria that's resident on your skin and some sort of issue with the immune system not being able to take care of that bacteria. We've also talked about keloid scars and this hyperproliferation that often results in your skin forming what is typically known as a benign tumor. So your skin actually has this capacity to overgrow, and that's a form of cancer. So this keloid scarring doesn't usually result in the development of squamous cell carcinoma, but it does show you that the skin has this ability to undergo this type of hyperplasia or overgrowth of tissue. So again, if you also take a look, and increasingly we're going to be looking at some of these other health-related issues. If you look at individuals who have ulcers associated with diabetes, so you often have diabetes resulting in skin ulcerations either on the feet or on the hands, etc. And in elderly patients, it tends to be overlooked where we get malignancies or skin cancers developing at these sites of chronic injury. So diabetes is a chronic disease. Aging is a chronic disease, if you want to think of it that way. Your skin is getting damaged, and in these individuals that have diabetes or that are aging, they also will have chronic skin diseases that may lead to malignant cancers of the skin. So all of this suggests that maybe there is this link between developing damage to your skin, having this chronic damage. This link with bacteria isn't really there yet, but I'm trying to make that case. But your skin, if it continually does get damaged, might lead to skin cancer or malignancy. And these are some of the examples that suggest that that's at least a possibility. So is it due to things like chronic inflammation. So we know, for example, your skin can overgrow. Those are those keloid scars. And if you take a look, there is another form of link or another link between skin wounding and the appearance of cancer. And one of these is more of a genetic sort of association. So in RDEB, which is recessive dystrophic epidermolysis bullosa, one of the things that happens is that in these individuals where their skin never fully repairs itself, and they blister their skin, it kind of heals, and then it gets damaged again. This chronic cycle that I've been mentioning with Marjolin's ulceration, this is a different example of this. This is also linked with a very high incidence of squamous cell carcinoma formation. So almost 100% of individuals with this genetic disease will develop at least one form of squamous cell carcinoma if they have this primary diagnosis. So again, there's a link, perhaps, chronic wounding, maybe something to do with chronic inflammation, because if you look at individuals that have this chronic wound on their skin, often it's also inflamed. And could this somehow be linked to developing this skin cancer? So again, one of the things that you should probably make sure that you are aware of are some of the different forms of proof, whether you're talking about Marjolin's ulcers or you're talking about RDEB. And by the way, if you are not comfortable with the entire spelling, if I've used an abbreviation in class and you want to use it on your short answer, you're free to do so, as long as it's an abbreviation that we've used in class or is a common abbreviation. So if you make up your own abbreviation and you come back later saying, but that's what I meant to say, that's not going to work. But if you use RDEB, that's perfectly fine as part of your short answer. And you'll understand what I mean when you go through the mock term test in your tutorial on Thursday. So again, it's probably a good idea to make sure that this slide is one that you go back and review, as well as this slide here, which goes into RDEB in a little bit more detail. So this form of bullosa is really dependent on where that skin is being affected. So if it's in the upper layers, in the epidermis, or if it's in the dermal layer, which is the RDEB that we've been mentioning, or if it occurs on the basement membrane, the form of this epidermolysis bullosa differs just a little bit. Again, we are talking primarily about this dystrophic form of the epidermolysis bullosa, and we're not going to be talking about the one that occurs at the basement membrane. That's the junctional form, nor the one that occurs on the upper layers of the skin, the epidermolysis bullosa simplex version, which is on the epidermal layer. We're concentrating on this dystrophic version that you see here. And this is what it typically looks like. These areas of the skin, and it's probably easier to see it on your own lecture notes. If you look at the individual's knuckles or the areas on the backs of their hands, they never really completely heal. So they always have sort of this redness, this inflammation, this sort of lack of healing that you see here. And this can lead to squamous cell carcinoma in these different individuals that have RDEB. Okay, so that's sort of the background. Now, I need to switch gears just a little bit. We're still talking about skin, but I need you to understand some of the mechanisms by which skin can become cancerous. And you need to know the different things that are on this particular slide, and it's important that you understand the Invalrukin model that I'm going to talk to you about in a moment. Okay, so do you have any questions about the background related to making a link between skin and the different forms of chronic diseases of the skin that we've been talking about, Marjolin's ulcerations, keloid scarring, or the type of RDEB we've just been looking at? All of them are associated with different forms of inflammation, skin damage, and eventually different forms of overgrowth or cancer. Yes, at the back. I didn't go into it in a lot of detail. Both of them involve like chronic damage to the skin. One is affecting a different layer of the skin, but we won't go into that in a lot of detail. We'll talk about it in the context of this paper and only in that context. Any other questions? So, yes? It's not due to damage. No, well, it's due to skin damage initially, but it's not due to any chronic form of damage. You can get it simply by having like a cut. Sometimes it can result from an allergic reaction on your skin, but no, it's not something that is chronic. It's usually not linked to malignancy, no. Any other questions? Okay, so I need you to now to kind of focus back on the mechanisms behind which this type of transition from wounded skin to cancer might actually occur. So if you look at wounded human skin, or you look at psoriasis of the skin or psoriatic lesions where you have like patches of your skin where it gets dry and flaky, for example, that would be a simple form of psoriasis. These type of lesions have been shown to be associated with an inappropriate expression of one of the molecules known as beta-1 integrin. So these, when you think of wounded skin, you automatically think of integrin beta-1, or if you think of psoriasis, you should be thinking of beta-1 integrin. And these are, again, extracellular matrix receptors that are in differentiating epidermal cell layers. So we have this overexpression of this particular extracellular matrix receptor, this integrin beta-1 receptor, in these different forms of wounds or psoriasis. So one of the things that happens when you have an overexpression or inappropriate expression of this extracellular matrix receptor in that area of the skin that has psoriasis or has inappropriate healing of the wound, one of the things that happens is that we will also get an upregulation in those cells that have this integrin beta-1. We will also get an upregulation of this extracellular signal regulated kinase, MAP kinase pathway, which is known as MEK1. So we get overexpression of integrin beta-1. One of the other things that happens in that area of the skin that has wounds or psoriasis is that we will also have the overexpression of this MEK1 pathway. So when you take this into account, you can actually develop an animal model for skin wounding, and this animal model might give you some insight into whether or not there's a link between skin wounding, where we have this integrin 1 pathway that seems to be so important, and the development of things like skin cancer. So if we have this MEK1, and when we talk about constitutively active, it means it's always turned on. You don't need a signal to turn it on. It's always turned on in a group of cells. And it happens to be under the control of what is known as the involrukin promoter. And this is known as an NVE. And I'll get to the EEs in a moment, but this is involrukin. This promoter is driving the expression of MEK1. Now, why is that important? Well, one, it's important because you need to tell me that you understand how this works on Tuesday. So that's the sort of practical element of it. The other reason why it's important is you need to understand that this promoter, this involrukin promoter, means that MEK1 is only going to be constitutively active in skin cells. This promoter only drives expression in skin cells. And guess what? When you overexpress MEK1, the constitutively active form of MEK1, the mouse that you generate will actually exhibit chronic skin inflammation, and it will also have epidermal hyperproliferation. It will develop tumors, in other words. And these are usually benign tumors. This hyperproliferation is usually benign, and these benign tumors, these little growths, like the growths that you might get on your skin related to a skin tag, like if you know someone that has diabetes and you see these little growths on their skin, these benign tumors, which are known as papillomas, are evident in these mice that have an overexpression of MEK1 when you start wounding them. Okay, so these mice having overexpressed MEC1, which we typically see in wounded tissue, and now if you go in and take these transgenic mice and wound them, then their wounds actually undergo hyperproliferation, they form benign tumors or papillomas, and they have all of the hallmark features of what we would look at in chronic wound healing. Do you have any questions on this before I go on? So making the association between integrin beta-1 extracellular matrix receptors, we overexpress those in wounded skin or psoriasis, as well as in those same cells, we also have an upregulation of this MEC1 pathway, really important. If we put that in a mouse, this mouse, when you wound it, will also develop chronic inflammation, papilloma formation, et cetera. So we can model wounding of the skin and hyperproliferation in these mice. So this is sort of the INVE mouse model. So the INV, involrukin, the EE promoter system. And again, you don't need to worry about having to draw all of this out. I need you to understand this. So I can put this transgene in any cell type that I want. This is constitutively active. It means it's always turned on. Whatever cell I put this gene into, it's going to be constitutively active. Why is it constitutively active? If you go back to the previous slide, it says INVEE. And that's because these two serine residues are converted to the glutamic acid residue, or the E, that you see here. So this makes it constitutively active. It's always turned on. And that means that whatever this promoter is driving in whatever cell type, this will make the MEC1 be expressed in those cells. Okay, so do you have any questions on this before we go on? Like any questions at all? It doesn't matter, like if you have anything on your mind, just let me know. Or if you want to talk during the break, let me know as well. You need to know this model system. You don't need to be able to draw this out, so I won't ask you to draw this structure out or anything else, but I need you to understand why we're talking about this. So the involrukin I haven't talked about yet. We've talked about this constitutively active MEC1, and I need you to understand that MEC1 is usually associated with wounded skin cells, and we're going to talk about why that's important. So if you have no questions, I'm just going to go on into why this INVE mouse model is so important. So what is involrukin, you might be asking? So involrukin is a gene product that is specific for keratinocytes. So where do we find keratinocytes? In our skin. So we have involrukin being expressed specifically in keratinocytes. Therefore, if we have the gene or the promoter for involrukin, it will drive the gene product that follows after that promoter. So how does it get turned on in the first place? So if I deliver this gene, there's that promoter, but how do I turn that promoter on? In the same cells, in the keratinocytes, there is another protein known as loricrin, and this is also produced by the epidermal keratinocytes, and this usually drives the expression. It binds to the promoter region and then drives the expression of the involrukin gene. We have replaced the involrukin gene with MEC1 in these animals, okay? And so when loricrin binds to the promoter, it turns on the promoter, but instead of turning on involrukin, now it's going to turn on MEC1. And MEC1 is always going to be turned on within those skin cells. So again, just making sure we're all on the same page, MEC1 is now going to be driven or going to be expressed constitutively in all the keratinocytes that you have in your skin. So loricrin is only produced by your skin cells, the keratinocytes in your skin, and we have this INVE promoter system to drive MEC1. So that means that MEC1 is only going to be driven to be overexpressed within those keratinocytes within your skin. And in fact, if you take a look, we do indeed get the formation of these papillomas. We get the expression of chronic hyperplasia, et cetera, within these animal models. So they have these little bulbs of skin that we can then section, and we can actually see that we have an overgrowth of skin just as we would expect within these mice. Okay, so I'm hoping that you understand what the INVI model system is, why we might use it, and I'm going to go through that model system in a little bit more detail in the next few slides, but I need you to make sure that you understand why this model system is important for skin and driving a marker that we would normally have in wound healing and the skin. So if it's not clear for you, feel free to ask during the break or during office hours next week or in tutorial as well. But you do, again, no lies, you do need to know this for your upcoming term test. So that's one aspect. So we are looking at a mouse model, and now we're looking at something else that we need to consider. So I've also mentioned that one of the things that happens in skin cancer is that there might be an interaction with the immune system. So we talked about that with anesthesia and the lack of neutrophil activation. We talked about that with immunosuppressants and the development of skin cancer. So your skin could theoretically be affected, and skin cancer could also arise because of a change in the immune system. So Fiona Watt's group is, again, kind of out there a little bit. So if you've learned about this in your IM 250 class or in other IM classes, that's great. But she's a little bit out there in terms of her model system that she's looking at. And one of the particular type of T cells that she's interested in are these peripheral gamma delta T cells. And these gamma delta T cells that you are seeing here have some unique properties that are associated with them. And they're believed to be responsible for the uncontrolled cell growth. So if you have some kind of aberrant signaling of these gamma delta T cells, it could result in uncontrolled cell growth or the development of tumors or cancers, at least according to Fiona Watt's group. So these are the different gamma delta T cells. Again, you don't need to worry about all of the things that are on this particular slide. However, if you were to take a look, one of the things that these types of T cells respond to, just down here at the bottom, is that they will respond to microbial antigens that are presented to them. So where do we have them? We have microbial antigens, for example, on our skin. The other thing I'm going to draw your attention to on these gamma delta T cells, the other thing that makes these immune cells kind of interesting for us in this particular context in this paper, not only do these react with microbial antigens, but in addition to that, they have very specialized toll-like receptors that are found on their cell surface. And these toll-like receptors, or TLRs, are ones that we need to keep in mind when we talk about some of the results of this paper as well. Okay, so we haven't really gone too far into this yet, so it's just something to keep in mind in terms of the immune system. These are cells that have been proposed to check aberrant cell growth. It might halt the development of tumors, and we haven't really looked at them yet. I do need you to know a little bit about what these gamma delta T cells do, and you do need to know the information as I presented it to you in lecture today. None of the other details on this slide are ones that you need to worry about. So again, going back to Fiona Watt's paper, what is this link between having chronic inflammation? So now that we have this INVI model, if you go back to one of the bullet points at the very bottom, this INVI model system has chronic inflammation. So we have a model system. So we can also damage the tissue. There's a wound modeling system that we can do in this mouse model. And then can we see the development of things like skin cancer? And is there a potential link between doing all of those things and the development of a bacterial response that leads to all of this? And can I prove that to you in Fiona Watt's paper? So you are never going to be asked to tell me about specific data. I want you to understand some of the data that I'm going to be presenting to you here. So if you look at the wild-type mouse, or you look at the in mouse is overexpressing MEK1 only in the skin cells. One of the things that you will see is that if you look at the number of papillomas, the tumors that develop, and you look at the overall wound size, the bigger the wound is, the bigger the papilloma that results in these in mice compared to the wild-type mice. And again, that's the only thing that I want you to take away, that when the bigger the wound is in the in mice compared to the wild-type, they get much more aggressive, bigger papillomas, those benign tumors. So you damage the skin, you get bigger benign tumors that result in the in mice. Interestingly, the other thing that you can make note of, you can mark immune cells that will migrate to the area of skin damage by marking them with a marker like CD45. And one of the things that you will see in these particular mice is that you will have immune cells infiltrating into the area of wounds, because you wound both the wild type as well as the in mice. But we get many, many more of these CD4 positive cells in the in skin system even before we get wounding. So we already have much more activation of these immune cells, these CD45 positive cells, and particularly after we do this wounding. So this suggests, because these are responding to inflammatory markers that are being released at the site of injury, that when you look at the in mice are always much, much higher in terms of the number of CD45 cells. So the white bars are always bigger, more CD45 cells. And the bigger the size of the lesion is, the more of these immune infiltrates that you get. It suggests that there's a link between immune cell migration and the formation of these benign tumors. So again, nothing all that surprising based on the hypothesis that we've been working on, but it's a really good model system to suggest that the in system, where we have MEK1 overexpression in the white bars, is indeed one that we could use to try to understand what's going on here. So going back, what are some of those inflammatory markers? And I do not need you to memorize the entire list of things that are over here on the right. These are, by the way, log values, so it doesn't look all that impressive. So if you look at, wow, it's not compared to wild type. So if I normalize and then compare these to wild type, wow, it doesn't look like a molecule like mid-88 is all that much higher. But just keep in mind, if you look at the y-axis, this is actually a logarithmic value, log base 2, and you're actually looking at quite a substantial increase in signaling. So when you compare this to the wild type, after you have damaged the skin, one of the things that seems to be really evident is all of these different downstream markers that you see here can be related back to NF-kappa B, or nuclear factor kappa B, in the inflammatory pathway. And of all of the different types of genes that were examined, the ones that I'd like you to pay the most attention to are things like tumor necrosis factor, alpha, interleukin-6. These were much higher even in tumor-free in mice, and even higher still in tumor-bearing in animals. So this suggests that there is an inflammatory pathway that is turned on by the NF-kappa B, and this is actually in the blood. It's systemically or found within the blood of these different animals. So there is chronic inflammation. There's absolutely no doubt that this inflammatory pathway is actually linked. So I'm just going to go through the slides related to TNF. So if we think that TNF is that important, there are a number of different approaches that we theoretically could take. We could block TNF. So if it's elevated, maybe just bring it down. But we also just want to be able to address, is TNF actually involved in the pathway related to tumor formation? So TNF is well known in many different contexts, but particularly the pro-inflammatory pathway. And we know that its effects are actually mediated by it interacting with its receptors. So there is a tumor necrosis factor receptor 1 and a tumor necrosis factor receptor 2. So there are also knockout animals where we knock out both of these different receptors for TNF-alpha. There's nothing for TNF-alpha to directly interact with. So these mice that are deficient in both of these receptors are actually deficient and resistant to skin cancer. So again, suggesting in a different study that maybe tumor necrosis factor might be important in signaling. Again, the skin cancer that they were looking at there was direct application of a chemical carcinogen on the skin. So when you put a carcinogenic chemical on your skin and then you wait a little while to see if it develops cancer or skin cancers, these mice that didn't have tumor necrosis factor receptors didn't develop those skin cancers. So going back to this paper now, so that's sort of an aside paper, not directly related to this one. When they looked at interfering with tumor necrosis factor receptor signaling, but in the white blood cells, in the leukocytes. We're not talking about skin here. We're talking about manipulating and blocking tumor necrosis factor receptor at the level of the bone marrow. So only the white blood cells are going to be tumor necrosis factor receptor deficient. And they are generating these tumor necrosis factor receptor 1 and receptor 2 knockouts. And these are deficient within the bone marrow. And they are crossing these with the in V mice. So the in V mice normally form tumors. And now these white blood cell, their white blood cells don't have the tumor necrosis factor. One of the things, again, surprisingly, by knocking out the tumor necrosis factor receptor in the white blood cells, we're not talking about throughout the whole animal, in the white blood cells, if you look at the normal progression of things like papillomas, so skin lesions or these benign tumors after you have wounded them. So let's say we wound them here at day 10, we wait a little while, then we get tumor formation. But in the knockout animals, we actually don't see that. So it's like it's blocked, or the papilloma formation has been blocked, and they are highly resistant. They're all the way back down here at the bottom where we don't get these papillomas. So simply by knocking out tumor necrosis factor receptors, so in the white blood cells, and these white blood cells, on that previous diagram, I showed you migrate to the areas of these wounds. By blocking that migration to these areas, we have actually prevented papilloma formation. A little contrary to what you'd expect, like don't immune cells actually block cancer? Well, in most cases, yes. Fiona Watt is controversial because she believes that some of these leukocytes are those specialized gamma delta T cells. And she believes that their migration into a wound site actually begins skin cancer. So opposite to what you might have thought the immune system might be doing. So I'll just take you through the one last slide, and then we'll take our usual break. So this tumor necrosis factor ablation in these leukocytes did definitely protected these mice from developing tumors after wounding. So the classic model, wound them, get these massive papillomas. The bigger the wound, the bigger the papilloma. When you knock out tumor necrosis factor, though, even though the in V mice overproduce this inflammatory response, without those receptors on the leukocytes, there is no tumor formation. And there's less direct infiltration. So that means that we don't have the migration of these gamma delta T cells into the area of the wound, unlike what we see in the normal in V mice. The normal in V mice with these massive tumors have these infiltrative gamma delta T cells, but we don't when we knock out the tumor necrosis factor receptors. And again, it seems that this selective reduction in these CD4 positive cells, the marker for the gamma delta T, so if we look at all the immune cells, we can use a marker like CD45. If we're looking specifically for these gamma delta T cells, one of the markers that we can use is this CD4 positive marker. We don't see them infiltrating as much. So suggesting that this is what causes this tumor formation. I know that's a lot to think about. You don't need to worry about being able to reproduce or label any of these slides. These are just there in case you're interested in kind of going through the science. And we'll take our break now. We'll hope that there is no fire alarm or anything else going on. So we'll come back in about 10 minutes. We'll go through the rest of the lecture. And again, I'll point out what you need to know along the way. Okay, folks. So if you don't mind, we'll get going. Just so hopefully I can end just a little bit early so that you can get home just a little bit earlier as well. So before we get back into skin cancer potentially and go back to some of those different immune cells, again, potentially, I need to take a little bit of a side track a little bit. And one of the things that we saw was that NF-kappa B was elevated overall within that in V mouse model system. Some of the things that are associated, some downstream, some upstream of this NF-kappa B pathway that you see here are things like the inflammatory cytokines, things that I mentioned that you should probably make sure you know, like tumor necrosis factor alpha, interleukin 1, interleukin 6, etc. But what actually is the importance of that? And again, going back to what you might have learned in either first or second year bio or in one of your IM courses, and just as a review for you, and you don't need to worry about all of the details on here. If we're taking a look at a particular cell, whether this is an immune cell or whether this is a different type of cell, for the time being, we'll just not worry about that differentiation. We have these different sorts of pattern recognition molecules that we have. So different types of bacteria, for example, might have different lipoproteins that these toll-like receptors are designed to be able to identify. Or they might have, for example, double-stranded RNAs, or they might have lipopolysaccharides that will interact with TLR4. But I'm drawing your attention to the one right here in the middle, which is TLR5. And TLR5 is a pattern recognition molecule found on some cells that we'll get to in a moment. And when these cells that have this TLR5 molecule, this toll-like receptor molecule on it, it will then initiate this cascade intracellularly inside the cell. So again, this would be the cell intracellular space. It would activate MYD88. It will cause the IRAC TRAF6 pathway to be turned on, and it will in turn activate that NF-kappa B pathway and lead to the transcription and production of different inflammatory cytokines from this cell type. Now, why is TLR5 important? TLR5 is important because it is the molecule that will recognize flagellin. And flagellin is this characteristic protein that is found on bacteria that have the flagelli. So when you see swimming bacteria, they have these flagelli, and this flagellin molecule that makes up the flagelli on bacteria is usually recognized by the immune system using this TLR5 molecule. So you need to recall those details before I can take you through the rest of the paper. So hopefully that's a bit of a review. You do need to know this slide, not the other molecules that I haven't talked about, but the TLR5 as well as the MYD88 pathway, et cetera, that links some of these concepts that I've been going through. You, again, don't need to worry about the structure of these different toll-like receptors. If you're interested or if you happen to have covered it before, this is sort of the classic structure of these different toll-like receptors. So why are we talking about MYD88 and TLR5? So if we are trying to make this link between wound healing, chronic wounds, the immune system, and its activation, we've done a pretty good job up until now. We haven't really talked about, do bacteria actually initiate the formation of these different tumors? And we haven't mentioned that at all. All we've been looking at so far is you get this in V mouse, great mouse model, constitutively active mech. When you wound it, the bigger the wound is, the bigger the papilloma that results. It makes perfect sense. And if we take a look, the immune system might be involved because if we eliminate the TNF receptors that are found on the leukocytes, the white blood cells that migrate into the areas of the wound, then they don't develop these tumors. But what is the link with bacteria? Why do we need to worry about this bacteria? So if you look at this MYD88 and TLR5 system, again, the MYD88, the myeloid differentiation primary response gene 88, is one of the innate signaling molecules that is related to all of these different toll-like receptors, the things that are designed to screen and to surveil all of these different potential pathogens that you might encounter. So going back to the same types of experiments they did with the TNF receptor mice, they were looking at whether or not this MYD88 was actually important. If it's part of that signaling pathway that leads to chronic inflammation, and we know NV mice have chronic inflammation, what happens if you disrupt this classic pathway that I just showed you, where this MYD88, as part of this TLR pathway, and they all converge on this pathway, what happens if you knock this out? You would hypothesize, as the authors did, that if this is important, and if this is important for sensing on the immune cells, the immune or the inflammatory pathway, by knocking out in the white blood cells, through knocking this out in the bone marrow, you would hypothesize that you would see exactly the same type of response that you did when you knocked out TNF receptors 1 and 2. And so by doing this in this NV mouse background, and this is the panel that I want you to pay attention to, by knocking down this MYD88 in the white blood cells, the ones that migrate to the area of skin damage, in fact, they saw that tumor sizes and the number of tumors were reduced just like they saw before with the TNF receptor knockouts. So these white blood cells seem to be important, and by knocking down the key signaling molecule, this led to the same sorts of results that they saw before. So without TNF receptors, they can't home into the site of damage, and without being able to activate their normal immune response downstream, you can also affect tumor formation as well. So is this due to any one of those different potential toll-like receptors? So if you, again, are a really big-name scientist and you have the time, money, postdocs, and everything else to be able to do this sequentially, which is what they did in this particular paper. They went through and they knocked out every possible toll-like receptor that they thought was reasonable. The only one where they saw a response that was a reduction in tumors. The only one where it doesn't look like a normal NV mouse, so every other graph, they kind of line up with each other. The only place where they saw a reduction is when they knocked out TLR5. So when you knock down TLR5 in the NV mouse background, one of the things that you see is the papillomas, the tumors, are also reduced. So this suggests that somehow in these immune cells that are known to migrate to the site of damage, that they are using this toll-like receptor 5 pathway. And if we eliminate that, we eliminate the formation of these benign tumors or these papillomas. So again, suggesting, I haven't shown and proven to you yet that it's due to skin bacteria, but this TLR5 through this MYD88 pathway on these white blood cells seems to suggest that bacteria might be involved, because going back to this previous slide, what does TLR5 normally interact with? It's the flagella or flagellin, the protein that is found on bacteria. Okay, so this is sort of a prelude to what we're trying to get to. Any questions on that before we go on? So it's been a step-by-step process, but now we're going to try to get to the link to these different bacteria. So flagellin, again, I'll abbreviate it sort of as FLA. You can do the same as long as it's something that we've covered in lecture. This is also, by the way, I pointed out you should know the MYD88 pathway. You should know what you would expect to happen to papilloma formation in an in vivo background when you alter the leukocyte expression of things like MYD88 or toll-like receptor 5. If I throw something in there like toll-like receptor 1, just ignore it. It's not something we looked at, but it's just there as a distractor for you. It's really homing in on TLR5. And the reason why we're interested in TLR5, it's the only known ligand for the toll-like receptor, the one that we just knocked out. And it's normally found on bacteria that have these flagelli. So if you think a little bit about this, one of the easiest ways to go back and test this hypothesis is mice, just like we do, have bacteria growing on their skin. What if you reduce the amount of bacteria on their skin? So if this is really due to the fact that somehow with wounded skin that has like overproduction of the in the pathway MEK1, would you actually reduce the size of the tumors if you actually didn't have as much bacteria growing on it? So mice were treated, as you would suspect, just knock out all of the bacteria. So you give them a broad spectrum antibiotic. And again, this is enrofloxacin. You can, again, abbreviate it as ENR if you want to. It's put in their drinking water, so you drink it, or you apply it topically on your skin, like you apply it directly on their skin. They all had this decreased skin bacterial load. So in these mice, going back where you have wiped out the bacteria, nothing else, you still have the NV mice. You now wound these NV mice. Their tumor sizes are actually indeed reduced. So this wound-induced tumor formation was greatly reduced. Tumor size was greatly reduced simply by using an antibiotic. Again, that's sort of the link that we've been looking for. Is there a link between bacteria and skin damage and the formation of a tumor, which again is a form of cancer? So again, if you think about some of the controls that you might look at, so methicillin is a very narrow spectrum antibiotic. This again targets only gram-positive bacteria, but this targets bacteria that's also abundant on the skin. So again, it's a really great control because of one issue. This methicillin works against a number of these gram-positive bacteria on your skin, things like Staphylococcus aureus. So we've encountered Staphylococcus when we looked at the skin microbiome, and Staphylococcus epidermidis. These are great, but they don't actually have flagelli. So you're knocking out all of these gram-positive skin bacteria, but you actually have no effect on tumor size when you do this type of antibiotic. So this suggests, again, that flagellin is playing a key role, that it is indeed skin bacteria of a specific type that can actually initiate tumors. Do you have any questions on this before we go on? And again, being completely upfront with you, because I know some of you get really stressed before a term test, this is a good slide for you to make sure that you know. Some of you have had other health and disease courses, or some of you have had access to different antibiotics, but this is a good one for you to go back and review. Yes? So when you think germ-free mice, what are you typically thinking of? But probably you're thinking about the gut microbiome. You're usually not thinking about the skin microbiome. And you have to also consider this, and sadly I don't have time to go through all of this. When you're taking antibiotics, typically we think about it only affecting your gut microbiome, but it's also affecting your skin microbiome as well. And germ-free mice, those are, again, a little different, but they are bred not to have a gut microbiome. We don't actually affect their skin microbiome. It's a good point, though. Any other questions, comments, thoughts? No? So again, this suggests, to tie all of this together, all of these data suggest that on your skin, you have a number of flagellated bacteria that are going to interact with immune cells that are part of your skin immune system, especially those gamma delta T cells, if we believe that Fiona Watt is correct, and that on these gamma delta T cells, we have these toll-like receptors that will be able to interact with these. And this is what is actually going to promote tumor formation in certain individuals, not everyone, in certain individuals that would potentially have like overexpression of things like MEK1. So who would have that? Someone that has chronic skin damage would have lots of integrin beta-1 receptors. It would have a lot of MEK1 activity. And those are the individuals where if we have these gamma delta T cells in the neighborhood, might actually cause the initiation of tumor formation in those specific cases. And again, don't worry about all of the details here. It's just some more background experiments that were done to actually prove that it is TLR5 in this background working specifically with flagellated bacteria to cause skin cancer. All right, so I'm hoping that this in-V mouse model system, some of the key components, how we actually go about determining that some types of bacteria on your skin could potentially cause cancer, all of those are ones that you will be able to tell me about on your term test on Tuesday. How many of you, by the way, intend to study on Saturday or Sunday? Ooh, is there another test going on? What else is going on? CSB329. How many of you are in CSB329? Not as many as I thought. Any other term tests going on? Yes. Oh yes, PSL 301. How many of you are in PSL 301 where you have it on the Monday right before this one? Okay, again, not as many as I thought. If you guys are okay with it, because I know some of you might be studying other things, I will probably put together sort of a Google Doc that you can ask questions on directly live on Sunday evening. So maybe around 8 o'clock. So if you have any last minute questions, or you have any questions, we'll do it in real time on Google Docs. I'll put the link up on the announcements page. You can ask questions there. I'll answer you in real time. I'll be online for maybe half an hour to 45 minutes, and you can always post questions ahead of time as well. And that will also, maybe I'll stick a few surprise questions on there as well for you that you can think about before Tuesday. Okay, so I do need to get through a few more slides for your term test. So I'm going to switch gears a little bit. And one of the things that I want to try to convince you of, health and disease, human biology is changing so incredibly rapidly. And one of the things that's really exciting is that we can take skin, wherever that skin source is, and we can actually generate what are known as organoids. And these small organs that are derived from your skin are eventually what we're going to. So we've been talking about skin. And one of the most interesting places outside of the kidney cells or the kidney organoids or the gut organoids that have been developed are the brain organoids. And the reason why we're so interested in the brain organoids is if we take skin from an individual that has been diagnosed with a specific disorder, where we don't really have good animal models or animal models that everyone can agree with, we might be able to actually go back in and figure out more about the disorder using these skin-derived organoids, and particularly something very, very complex, because no one can agree on this, something like schizophrenia. So I'm going to take you through really quickly today what schizoid type disorders are, or what schizophrenia resembles, what we currently know about schizophrenia, and do we actually know a lot about the genetics of schizophrenia very, very briefly, and we'll come back to that when we go through the next paper associated with skin organoids, and then how we can build this model system that we're not all in agreement about. So this is the most classic model that I'll take you through just as an outline, and then maybe suggest that we need to rethink how we look at these very, very complex disorders or these different diseases. So again, this is somewhat controversial. You probably have taken other courses where some of the things that are on here may not be in complete agreement with what you have learned. So one of the things that I'm going to be talking about is the fact that schizophrenia or schizoid type of disorders is genetically linked. I won't say heritable, because sometimes someone in your family might have schizophrenia or a diagnosis of schizophrenia, and it doesn't mean that their offspring will have schizophrenia, but usually there are some other mental health-related concerns or other types of mental health disorders in those individuals. So there's a linkage to schizophrenia that involves some sort of heritability, but it doesn't mean that schizophrenia is going to be passed down. So this usually involves impairments in cognition. And we have this really biased view of what schizophrenia is. So those individuals that you might see on the streets that are shouting or seem to have these hallucinations or are really angry and upset, that's sort of a very stereotyped view of it. The thing that bothers schizophrenics the most is really problems with thinking, and it's disordered thinking, not being able to concentrate. Cognition is actually the #1 sort of deficit in these individuals. They have problems with perception. They don't see the world the same way that we do, and their motivation seems to also be really affected. Usually, this undergoes what is known as a sort of prodromal period. This usually occurs in adolescence. We see this typically in males in their adolescence. It's a little later in females. And in females, it's more likely to appear in adulthood. And these signs and symptoms that I'll get to in a moment. We don't really know the pathogenic mechanisms. It's a cause for great debate. Like, what is the cause of schizophrenia? Is it really genetic? Is it genetic and environment? Is it something that happened during early development? Is it something that happened in certain groups of cells? Is it neurons? Is it glia? And there is no consensus on what these pathogenic mechanisms are. However, there is one factor that, regardless of what theory you believe in, is that there is a loss of gray matter. So there's a loss of neurons in certain areas of the brain that we'll be talking about. And we see that routinely in individuals that have schizophrenia. And also very commonly observed is a reduction in the number of synapses or connections between neurons in individuals with schizophrenia versus age-matched counterparts. So there's something definitely going on within the brain of these individuals. You might think that there are some great pharmacological treatments, and there are. Some of the symptoms of schizophrenia can be controlled or tolerated using different types of pharmacology, but they're not universally well adapted for everyone. And again, I don't know about you, but when I was doing my undergrad here, so I started off actually in pharmacology, toxicology, and one of the things that we were always taught in our courses was that you always needed to have a receptor, and the drug had to be able to be shown to bind to it, and you needed to understand the mechanism for this to be considered like proper pharmacology. We don't really have a classic mechanistic understanding. Like, we can give these drugs that make it look like these individuals that have schizophrenic symptoms, they get better, but we actually don't fully understand how these drugs work. We don't really have good drugs to actually improve that cognitive impairment. When they can't concentrate, we can help to manage their hallucinations. We might be able to help with some of their negative symptoms, but their inability to concentrate, we don't have good drugs to be able to help them with that. And again, we don't know how we can affect all of the different signs and symptoms of schizophrenia as well. Again, there's a lot we don't know. Even the genetics, we're not 100% sure of this. So when you talk about schizophrenia, again, we don't talk about schizophrenia. It's a scary disorder. But think about this statistic. We think that it's about 1 in 100 individuals that get affected with schizophrenia. That means that, for example, on campus where we have thousands of students, it probably means that there are many individuals that have schizophrenia. And the average age of onset is about 18 in males. It's a little bit later in females, 25 years of age. And so, you're being exposed to individuals that are 18, 25 on campus. And it means that we need to take this disorder very, very seriously. And there are a lot of issues because of the differences in age. So it seems like 7 years, not that much of a difference between males and females, but this age gap as well as this gender difference actually does make it challenging to identify some of the symptoms that we typify within what is known as the prodromal period, the milder period before the full-blown symptoms can actually occur. So how do we find that early before they start hallucinating wildly or having some very unwanted thoughts or actions coming into play? So again, these cognitive changes, again, maybe not surprising. You might know individuals that have schizoid type disorders. There's trouble usually at school or at work. Again, not surprising looking at the age range of individuals that are affected that we would be looking at school or work. Oftentimes this means that they have difficulty concentrating or thinking clearly, but most of the time, when you think of schizophrenia, it's the ones that are highlighted for you, that these individuals are suspicious or they can't trust other individuals, or typically they hallucinate, or they have a very different sort of perception of what is actually going on around them. And oftentimes, this will lead to things like withdrawal from friends as well as family, and it also means that they're stopped taking care of themselves. If you look at the most striking feature that's associated most commonly with schizophrenia, what is sort of the hallucination that many of you have learned about? So what's the most common type of hallucination that schizophrenics or individuals with schizoid type behavior begin with? Any ideas? So it is a perception hallucination, yes. Auditory. And what's the most common auditory out of all of them? Sure, so thinking thoughts, hearing voices, all of that, but there's something actually even more common before all of those, probably in the prodromal period. Anything else? You're right, those are very typical symptoms like hearing voices in your head. Anything else though before that? The most common symptom? Schizophrenia? Where's Alexa for you now? No? So it's knocking. So they will hear sounds of knocking, and this is often the first sort of auditory hallucination. No one's actually knocking, but they keep hearing like a knocking noise or repetitive knocking-like sounds, and then they will gradually have more severe auditory hallucinations. So this gets into schizophrenia is such a complex disorder because if you look at some of the other types of disorders, like depression, you're always like in a down state, like you're down. That's what depression is. But schizophrenia is actually characterized by two different things that make it really hard to understand. Even as a neurobiologist, it's really hard for me to understand how these different behavioral outcomes can actually coexist in one brain. So we have both positive symptoms associated with schizophrenia. So these are hallucinations, whether they're auditory, like you think you hear something when something's actually not occurring, or you think you see something when actually something's not there. It could be delusions. It could be you have disordered thoughts, like you're not thinking rationally. So I think it would be a good idea for me to put my hand on this burning stove because I think that my hand would smell good. Disordered thinking. Again, not something rational. And then we also have negative symptoms. So that's the one side, and that's the side that we think the most about with schizophrenics. But when we look at negative symptoms, there's also like a shutdown of different areas of the brain. So there's alogia, so the inability to verbalize or to talk. There is withdrawal. They don't want to socially engage with individuals. They generally have apathy. So on the one hand, your brain is doing stuff that it shouldn't be doing. And on the other hand, your brain is also not doing things that it should be doing. So how do you get those two different things to work in the same brain? And of course, cognitive deficits. And if you're thinking about this based on whatever you've learned in your psychology courses, this means pretty much every system in your brain is affected in different ways by, if you're looking at all of these different symptoms, like all of these things involve different circuits within your brain, and they're not all going in the same direction. It makes it a bizarre and very interesting, very challenging disorder. And again, how do we model this? We don't have a good animal model system. So again, what we try to do in biology is to develop a model system. So what I've just spent the last week and a half is taking you through a model system where we're trying to understand cancer in the model of a mouse, and I try to get you to understand how that model system was developed. And when we look at how do we understand schizophrenia, it's based on model systems that we've developed over the years. And one of the things that is very commonly associated with schizophrenia is things related to an aberration or something going wrong with the dopaminergic system, whether it's too much dopamine, which is the most common theory, overactivity of the dopaminergic system, and this gets into the pathophysiological hypothesis of schizophrenia that's dominated over the last 50 years, that only in the last 5 years we've started to rethink. And some of this is based on experimental data from animal models, some of it's based on observational studies in humans, and there's absolutely no doubt that the dopaminergic system is going awry, but we don't really understand why it's going awry. If you look at some of the different agents that will overactivate the dopaminergic system, there are many different drugs that will actually do this, especially different psychostimulants like amphetamine. These psychostimulants, if you are taking them for prolonged periods of time or you're taking them at high doses, actually do produce a psychosis, which is, again, like a hallucination or like changes in thought and sort of disordered thoughts that are associated with this. And this is in healthy individuals. And even worse, if you take an individual who has been diagnosed with schizophrenia or is undergoing that prodromal milder symptom period, and they're given a psychostimulant that involves overactivation of the dopaminergic system, their symptoms, their hallucinations, their inability to concentrate actually become much, much worse after taking this dopaminergic agonist or something that stimulates the dopaminergic system. And again, just taking a look at this from a systems point of view, and you do not need to memorize all of this. None of this diagram will be on your upcoming term test. It's just to give you sort of a broad overview. If you were to take a look at the role of some of these different dopaminergic systems, there's a very limited number of dopamine-producing neurons within your brain. Okay, so we don't have a lot. It's not like glutamate. Glutamate dominates your brain. GABA, we have a lot of different GABAergic neurons. But we don't really have a lot of neurons that make and release dopamine. There are probably around 5 or 6 different nuclei that will do this. One of them, which is highlighted for you here, is known as the ventral tegmental area. The other one is the substantia nigra. But again, those names not being really important, but these circuits, this dopaminergic neuromodulator, it's actually not a neurotransmitter. It modulates the activity of neurons. One of the things that it does is it will change motivational status. So by changing the levels of dopamine, you can change like your motivation to finish a task or not to finish a task. It will affect your motor pathways. It will also make things more pleasant, or it will give you more pleasure, and so it is associated with addiction pathways. And again, these are some of the signs and symptoms that we see in individuals with schizophrenia. They have movement disorders affecting motor pathways, they have a lack of motivation, etc. So there's a lot to suggest that maybe the cognitive deficits, some of these other signs and symptoms are affected in schizophrenia. If you know anyone who has taken a high dose of L-DOPA, you'll know that they also have some behavioral changes that look a lot like schizophrenia as well. And L-DOPA is the precursor to dopamine. So you have too much L-DOPA, meaning that you'll produce too much dopamine. So again, this is a really busy slide, and I don't need you to memorize all of it. And I only want you to take away 2 different things from this particular slide, and I have only a few more to get through, and that's all you need to know for the upcoming term test. We're going to come back to schizophrenia and brain organoids to model schizophrenia later on in the course, so you don't need to worry that you're going to miss out on anything. We'll come back to it. But there's a couple of things on this slide that I want you to know, but you don't need to memorize everything. One of the things that seems to be really important is that dopamine can interact with receptors that are found presynaptically. So this is the presynaptic. It's going to release the neuromodulator dopamine. You can always tell the presynaptic compartment by looking at these vesicles filled with a neurotransmitter or a neuromodulator, and here's dopamine packaged into that vesicle. And one of the things that we see is that dopamine can interact with this presynaptic receptor known as the D2 autoreceptor. So dopamine can interact presynaptically. Dopamine can also work postsynaptically. So we have receptors that are G protein coupled on the postsynaptic side. So dopamine kind of works in a variety of different places. Again, just sort of keeping that in mind. The other thing, as I mentioned to you in the previous slide, a lot of these different psychostimulants, drugs that we can take, things like cocaine, for example, one of the things that cocaine does is it will actually inhibit this dopamine transport system. And what that means is that in the presence of cocaine, this dopamine transporter, this DAT protein, is being blocked. That's why we have that minus sign there. That means that dopamine is staying out in the synapse longer than it normally should. So it's like increasing the levels of dopamine indirectly. The other things that I mentioned to you earlier as well is one of the things that we know, another psychostimulant like amphetamine. So amphetamine kind of looks a little bit like dopamine. So amphetamine is actually transported into the presynaptic terminal through this dopamine transporter. And because it kind of looks like dopamine, it gets taken up, but it actually isn't dopamine. So it poisons this system here, which is known as the VMAT system. And one of the things that this means is that it doesn't cause the internalization into the vesicle of dopamine. So dopamine just stays out and can get released much more effectively than it normally would. So these drugs have been used in previous studies to actually suggest that this psychotic event involves dopamine. So cocaine induces psychosis, and so does amphetamine. Both of these elevate dopamine. Therefore, if psychosis is one of the hallmark features of schizophrenia, this whole disorder must involve dopamine. And that's sort of the rationale that scientists used and tried to develop animal models to study this. Again, if you look at all of the different drugs of abuse, and nothing on this slide will be tested, so you don't need to worry about it. If this is a system that we think is being affected in schizophrenia, this dopaminergic system and that ventral tegmental area system that I mentioned to you before, most of the drugs of abuse involve things that will affect this dopamine signaling. And most of these drugs that you see listed up here will eventually cause the release of dopamine. And we've looked at the previous two, which you do need to know, amphetamine as well as cocaine, increasing the level of dopamine at the synapse. So again, this is just one part of schizophrenia. It may account for the positive symptoms. It's probably a good idea, by the way, when you're thinking about the short answer questions, if I were to ask you to name me 2 negative symptoms associated with schizophrenia, could you name me 2 right now off the top of your head? It's only been 10 minutes. So withdrawal and so alogia, all of those things. So again, those short answer questions are literally supposed to be like 1 or 2 words. So just keeping that in mind. And I think I'm going to just stop here because some of these studies will be important later on. And you don't need to worry about Wolfram Schultz for the time being. We'll come back to him. Let me talk to you a little bit about the term test and how you should be getting ready for this. So I'm going to tell you how I designed the term test and why it's going to take you a little bit less than 45 minutes to finish it. So you only have 15 multiple choice. I want you to have time to be able to go back over your answers. So these 15 multiple choice come from the 4 lectures, including today's lecture. It's actually dominated by the last 2 lectures. So today's lecture, there are multiple choice test questions. There are also multiple choice test questions that come from lecture 2. So 2 and 4 actually dominate the multiple choice test questions. The slides from tutorials. I have to ask because I haven't been talking to the TAs, not because I don't want to, but they're busy, I'm busy. just hasn't happened. Are the TAs giving you access to last week's tutorial slides? They are supposed to give you access to the tutorial slides. I will also post them for you, but please ask them. You're supposed to take your mock test home with you at the end of the day Thursday, so you get to practice it, you get to take up the answers, same level of difficulty, then you can take that home with you. It's, again, to help you study, but I will post those review slides for you if you haven't received them. You get 10 one-line or one-word responses worth 10 marks, so 15 marks, 10 marks, and then 2 short answers with parts. Some of this might involve like drawing, but it is designed to allow you to finish within about 45 minutes or so. In tutorial, you are going to be given practice questions, which will give you a good indicator of the level of difficulty, how specific the questions are. 1 or 2 of them might be very, very similar to what you get on the actual term test itself. So it is a great idea to show up to that tutorial, take that test paper with you. That is one thing I will not be posting. That's something that you'll take up with your group and your TA in class. And again, the only other thing, on the weekend, I will be posting a link to Google Docs where I will be live online with you. You can ask any questions. And to be honest, I'll probably post a couple of thought questions for you, or maybe even a sample multiple choice online at that time for you to go back and just see like how prepared you are for Tuesday. And I will also post office hours for Monday. So I know you're busy with other things. Please get started off on the right foot in this course. Get a really good mark on term test 1. I am making it completely fair. So you want a good mark to just get you motivated to keep doing well throughout the rest of the course. All right, so have a good tutorial. Look out for that link for Google Docs, and I will see you in the exam center next Tuesday.